Good afternoon, everyone. Thank you so much for joining us for our second webinar in our Plant Endophyte webinar series. I'd like to thank you all for attending. I wanted to first take a moment to introduce the Institute. The Institute for Advanced Learning Research has been in Danville since 2003, and our main goal is economic transformation. We do that in a lot of different ways, including a program in advanced manufacturing, where we show students the future of manufacturing and the most cutting-edge skills. We do that through advanced learning, where we have middle school students, high school students, and others come through the building almost daily. We have economic development in-house, and we also have wonderful conference services. But I'd like to take a moment to just go ahead and introduce our speaker today, Dr. Sajiva Amaradasa, and I'll turn it over to him.
Thank you, Dr. Lohman. Before I start my talk, I would like to introduce myself. I got my PhD in plant pathology from Virginia Tech. For my thesis project, I studied Rhizoctonia solanae. It's a soil-borne fungal pathogen. It has morphologically similar but genetically different groups. I developed molecular techniques to identify accurately these groups. Then I moved to University of Nebraska-Lincoln as a postdoctoral researcher to study Sclerotinia sclerotiorum. This is again, a soil-borne pathogen. It causes white mold in many economically important crops. Specifically, I studied if susceptible isolates are subjected to sublethal level of fungicide doses, could they become resistant by increasing their mutation rates? Usually, naturally existing resistant isolates multiply rapidly and become dominant and create resistance to a fungicide. But our research found that this particular pathogen, even susceptible isolates, can become resistant by increasing mutation rate over time if subjected to sublethal level doses of fungicide. Then I joined University of Florida, Gainesville, to study Fusarium wilt in watermelon. Seedless watermelons, this crop was introduced to the market in 1990s, so it's relatively new. However, it is highly susceptible to soil-borne pathogens. Farmers, growers used methyl bromide to fumigate their fields to suppress disease pressure. However, now methyl bromide is banned, so universities are studying different alternatives for methyl bromide. So I got the opportunity to test the hypothesis: if planting date is delayed by a few weeks, could we reduce the yield reduction by Fusarium pathogen? The hypothesis behind this was that Fusarium oxysporum prefers cooler temperature for infecting watermelon. Our study found that the reduction of yield was reduced by 10 to 15% when we delay the planting date by 3 weeks. You may be wondering why then farmers prefer to cultivate or to grow their plants early in the season, because in the USA, the highest demand for seedless watermelon is during the July 4th holidays, so they don't want to miss that opportunity. Thereafter, I joined the Institute in Danville, Virginia, to conduct their biocontrol program using beneficial bacterial endophytes. Before I start my talk, if you have any questions, I would like to invite you to use our chat feature and send those questions. We have allocated several minutes to answer your questions. And now I will begin my presentation. Today I'm going to talk about exploring the potential of beneficial bacterial endophytes for disease control. Plant Endophyte Research Center is a part of the research department of IALR. This is our team. Dr. Scott Lohman is the director, and he's a microbiologist and also an expert in writing research grant proposals and collaborating with industry and academia. Dr. May is the chief scientist. He's a plant physiologist and responsible for screening our endophytes for plant growth promotion. Robert Cretion is an expert in molecular biology. Amy Turner specializes in tissue culture, but she's also an all-rounder. Endophytes are microbial plant symbionts living inside plants. We have a large collection of endophytes. They are largely bacteria and easily culturable. In that way, we can do our experiments without any problems. This is the famous graph that shows world population over time. At present, there are 7.5 billion people on this planet. United Nations predict by 2050, there will be 10 billion people. So we are going to have 2.5 billion extra mouths to feed. Traditional method of increasing food production is to use high-yield cultivars and applying synthetic pesticides to prevent crop loss. However, synthetic pesticides are not safe for humans. They pollute the environment, lead to rapid buildup of resistance, and kill non-target beneficial organisms. That can result in secondary pest outbreaks. This graph shows the average research and development cost for a new crop protection product. In 1995, it was $152 million, but in 2014, the cost increased to $286 million. The average total number of products tested leading to a successful crop protection product also increased from 52,500 in 1995 to 159,500 in 2014. And also, the number of years between first synthesis of novel molecule and the first sale of the product also steadily went up from 8.3 years in 1995 to 11.3 years in 2014. These statistics indicate it is getting harder and harder to find effective and safe synthetic pesticides. Biopesticides are a great alternative for synthetic pesticides because they are safe, naturally occurring, and more sustainable. Plant endophytes are just one component of biopesticide market. Currently, biopesticide market is worth $3 billion of $56 billion global pesticide market. However, annual growth of biopesticide is 15%, and it is anticipated that in another 25 to 30 years, biopesticide market share will be equal to the synthetic pesticides. As I said before, our world population is increasing rapidly, and we need to find solutions, or we need to increase our food production in a more sustainable manner. Plant endophyte research is relatively new. We have so much to learn in this field. Their modes of action in plants are largely unknown. This graph shows there were hardly any peer-reviewed publications before year 2000. But in the recent years, we could see more and more research papers being published. Burkholderia phytoferments, strain PSJN, is one of the most studied bacterial endophytes. Strain PSJN is a biostimulant. Here you can see growth promotion activity of PSJN on switchgrass compared to the control. Dr. Jerzy Nowak of Virginia Tech, who is a pioneer in endophyte research, was instrumental in initiating this work. His team visualized the endophytic colonization of switchgrass after tagging it with GFP protein. In this table, you can see the number of colonies at different intervals after inoculation. At the end of 3 days, bacterial colonies were found mainly from roots. After 7 days of inoculation, the bacterial colonies appeared in leaf extracts, but to a lesser extent. However, 14 days after inoculation, more colonies were found in sheath and leaf tissue compared to the roots. This indicated endophyte PSJN not only could colonize the plant, but also could translocate within the plant. I don't expect you to read this slide. This flowchart shows how we established our endophyte library. We collect wild plants from different environments, such as nutrient-poor soil, flooded or drought-stricken lands for endophyte isolation. At present, we have more than 2,000 endophytes in our library. Isolated bacterial endophytes are characterized for growth promotion activities such as nitrogen fixation, phosphate solubilization, and auxin production. They are also screened for antifungal and antibacterial activity in vitro. We also have capabilities to analyze bacteria for production of antibiotics, such as surfactants and iturins. I have a collection of important plant pathogens to test our endophytes. I collected them from local areas, and some of them came from Virginia Tech Plant Pathology Department. First step in screening process is to do a plate assay like shown in this slide. The endophytes are rated for the level of growth inhibition. This is a quick method to screen a large number of endophytes. The promising endophytes are taken to the next level where we test them in planta. In this table, I have summarized plate assay results, though it is not comprehensive. The pathogens I used for the plate assay and number of promising endophytes resulted are shown. As I said before, this is the starting point of endophyte screening for biocontrol activities. Now let's look at some of the growth chamber and greenhouse experiments conducted with promising endophytes. Sheath blight caused by Rhizoctonia solanae is a major disease of rice. 3 endophytes from our collection were inoculated individually and in combination to test their biocontrol activity against this disease. We inoculated these endophytes by soaking rice plants in bacterial solutions overnight. After 1 week, pathogen was introduced and plants were rated for vigor and disease severity. The vigor and severity of endophyte treated plants were better than the control. However, endophytes in combination did not produce any significant results. Moving on to the next experiment, Pythium ultimum causes damping off in many economically important plants at seedling stage. We tested biocontrol of Pythium damping off in cucumber using 3 of our endophytes. Plants were inoculated with endophytes by adding 20 milliliters of liquid culture into the root zones. After 5 days, pathogen was introduced. Here you can see results of one such endophyte. The treated plants have 100% survivability, whereas the control plants, the half of the control plants are dying. This is just preliminary data. We are planning to do a much larger experiment soon. Root and stem rot of soybean is caused by Phytophthora soja. This is a soil-borne pathogen. Field symptoms are shown in the right-hand image. Pathogen infects the root system and wilts the whole plant gradually. The easiest way to see symptoms in the greenhouse is to inoculate stems with mycelial plugs, like shown to your left. This was an industry-guided experiment. This particular company wanted to test whether rare sugars galactose, mannose, and allulose can control soybean root and stem rot. We also added 1 potential endophyte from our collection as a standalone treatment and in combination with rare sugars. Results showed disease severity was not significant for any of the treatments. However, plant vigor was significant for endophyte as well as all 3 rare sugars. The best results were given for the endophyte 267, that's the endophyte we used in combination with mannose sugar. Now, let me give two examples of research guided by academia. Strawberry plants are susceptible to many soil-borne pathogens. Growers depended heavily on methyl bromide for soil fumigation. Since it is banned now, Dr. Jayesh Samtani of Virginia Tech approached us to investigate whether our endophytes could be used as a substitute for methyl bromide in controlling soil-borne pathogens. We tested 3 common strawberry pathogens against our endophyte collections. These pathogens were Fusarium oxysporum, Coledotrichum acutatum, and Coledotrichum gliosporioides. The best 3, after doing that experiment, the best 3 endophytes were selected for greenhouse studies. For greenhouse studies, we selected 3 strawberry cultivars. The strawberries were first inoculated with 3 endophytes by soaking in bacterial solutions. And after 1 week, Coledotrichum pathogen was introduced to the plants as a spore solution. All 3 endophytes increased total fresh weight and plant survivability of all strawberry cultivars tested. At the bottom, you can see endophyte 619, that is one of the endophytes we used in this experiment, had increased root growth of strawberry compared to the control. The endophyte 619 was further tested in 3 field sites. We depended on natural pathogen infection for disease and did not do any artificial inoculation. All sites treated with 619 had higher marketable and total yields. Marketable yield was 8.8% to 16% higher in endophyte treated plants compared to the control. The increased total yield ranged from 14.3 to 17% for 3 sites. We repeated the experiment again and got similar results. We are planning to submit these results to a peer-reviewed journal soon. Endophyte 619 was later identified as a Bacillus velesensis. Another example of collaborative research is tobacco black shank control. This is a collaboration with Virginia Tech Blackstone Research Center. This disease is caused by a soil-borne oomycete called Phytophthora nicotiani. We tested 2 endophytes in 2 field sites. The experiment design was a RCBD with 4 replications. Each treatment had 12 plants per plot. The planting holes were added with pathogen-infected grains to induce disease. In one site, endophyte 1835 performed better with higher fresh weight and vigor than the positive control. For the final part of my presentation, I would like to talk about several new initiatives undertaken by IALR. Controlled Environment Agriculture Innovation Center is a collaborative project with Virginia Tech's School of Plant and Environmental Sciences. It includes indoor and greenhouse hydroponic units. Dr. Michael Evans of Virginia Tech oversees this unit, and currently we are conducting experiments to investigate growth promotion activity of our endophytes in hydroponic plants. Another initiative is using smart tables for disease prediction and monitoring plant growth promotion. A smart table is a bench with a camera mounted on the top. The camera moves from plant to plant and takes an image at regular intervals. These images can be analyzed to compare movements of healthy and stressed plants. Usually, plants show stress signals before showing disease symptoms, and this can be used as a disease prediction tool. We are also collaborating with a producer of hydroponic lettuce in testing our endophytes against Pythium and Fusarium, which they find difficult to control using organic methods. We have already conducted plate assays and identified several potential endophytes to be tested in their system.
Okay, thanks so much for watching the webinar. I know we've had a couple questions come in, and Sajeev and I will be addressing those. Number one, I see a question relating to are we continuing to expand the Endophyte Library and what commercialization opportunities are there for them? So at the moment, we are not expanding our library. We found that our library has lots of potential as it is. It's a toolbox that does pretty much what we need. Of course, we could continue to expand it for decades, but we instead chose, because of limited resources and staff, to dive into what each endophyte does and better utilize the ones we have. We have a lot of commercialization potential for a lot of these endophytes, including working with companies that have specific needs. As you know, they are natural organisms, so they're relatively easy to get to market. And in fact, we're getting ready to publish a series of papers looking at both biocontrol endophytes and endophytes that help plants gather phosphates. So yeah, there's a lot of potential, but they are also very specific, and a lot of work still needs to be done, including knowing exactly what and when to apply the endophytes, the specific plant models. But that's why we have Dr. Meridasa here, is to develop pathoassays to tackle certain questions. And I'd encourage anyone that's interested to reach out to us directly through our Plant Endophyte Research Center to see if there are any collaboration opportunities. If there's some particular system or plant and pathogenic bacteria that you're interested in, please feel free to run that by us, and we can see if it fits into our model of working with industry.
Another question we got was, what future do you see for plant endophytes as biocontrol agents? Actually, I see a lot of potential. We have found several endophytes in our collection with broad spectrum activity against fungal pathogens. What we do not know is how these endophytes going to perform, what is their persistence in the plants. So we have to do more research, but we have these potential endophytes already identified in the lab. We are collaborating with industry to make them pellets and to apply them in the field and test them. It is a few years away, but I am excited about the potential of endophytes.
Are there any more questions? Okay, with that, I'll start to wrap up the webinar. If you have any more last-minute questions, please submit those. But I would like to thank everyone again for attending the webinar. It's something that we're going to do more of. We have a lot of different opportunities with bacterial endophytes, not only on the biocontrol side, but through mechanisms of action such as nutrient acquisition, through stress hormone level reduction, through ACC deaminase and others. We intend to have specific webinars on each of those, and probably even our imaging system that Dr. Meridasa mentioned during his presentation. So stay tuned for those, and we look forward to seeing you soon.
All right, thank you.